So today we will talk about functions of liver. So there are actually many functions of liver, and one of them is metabolism of carbohydrates. So in previous video I discussed about anatomy of liver, the structure of the lobes and all. Basically there are four lobes, two main, two are not main, like we have two main which are anterior, which are right and left, and caudate are not main, but they are also the lobes of liver. Now we talk about functions of liver. Basically the number one function of liver is metabolism of carbohydrates. Metabolism of carbohydrates. Okay, what are the carbohydrates? Like glucose. So what will happen, basically glucose, as we know that glucose is stored in liver in the form of glycogen. Okay, the whole mechanism we will discuss in the area of pancreas, which is my next video, right? So when glucose is converted into glycogen, this process is known as glycogenesis. This process is known as glycogenesis. It means glucose is converted into glycogen. This process is actually it is reversible. This is reversible. So when glycogen is converted into glucose, this process is known as glycogenolysis. This process is known as glycogenolysis, right? Now second, we have, if for example we need instant energy, for example if fatty acids, amino acids, or even the proteins break down into glucose, that process is known as gluconeogenesis. This process is known as gluconeogenesis. When we need, for example, instant energy, and our body will break down fatty acids, amino acids, protein, whatever it will get, and it will convert them into glucose, and the process is known as gluconeogenesis. Okay, in our body basically there is a ranking. Okay, first of all glucose will be break down, second glycogen will be broken down, third fats will be broken down, and number fourth is proteins. So proteins break down in very last. Okay, then we have metabolism of lipids. Metabolism of lipids, right? Now when lipid is converted into fatty acid and glycerol, this process known as lipolysis. Right? And when, if it is reversible reaction, it is lipogenesis. Lipid is converted into fatty acid, it is known as lipolysis, and if it is made like when fatty acid and glycerol combine together and they form lipid, that is or fat, that is known as lipogenesis, right? So then we are having metabolism of proteins. Then we have metabolism of proteins. What will happen, basically number first step is transamination. What does that mean? It means that amino group will be transferred from one amino acid to another amino acid. Basically, it is a transfer of amino group from one amino acid to other. This is known as transamination. Now second is deamination. What does that mean? In deamination, basically the amino group, which is NH3, will be removed from amino acid. Amino group, this NH3 group will be removed from amino acid, and then this basically ammonia will be formed. Ammonia will be formed. So as we know, this ammonia is very toxic substance. So then this ammonia will be converted into urea. The formula of urea is CONH22. So it will be converted into urea, which is less toxic substance. Which is less toxic substance, and the formation, like the conversion of ammonia into urea, is performed by a process which is known as ornithine cycle. Ornithine cycle, right? So the ammonia is converted into urea. Then what's another work? It stores some fat soluble vitamins. Like the storage is also work of this liver. There is a storage of vitamins. Which vitamins? Vitamin A, vitamin K, vitamin E. Some actually these are the fat soluble vitamins. Fat soluble, right? It also stores some water soluble, which are, for example, vitamin B with vitamin C. So it stores some fat soluble vitamins, some water soluble vitamins, both, right? And then it also performs a process of erythropoiesis. Now what is this? Okay, when like it at embryonic stage, this liver works as erythropoietic organ. Now what does that mean? Basically at embryonic stage, this liver forms the formation of RBCs is done by liver at embryonic stage, right? Now next we have, it works in detoxification. Now what does that mean? Basically, there are some people who drink alcohol. So alcohol is basically a poisonous substance, is a toxic substance. So this alcohol is detoxified with the help of liver. Actually, a organelle is involved in this, which is known as endoplasmic reticulum. Its smooth side is involved in detoxification, right? Now next, what are the functions of liver is so it also works in thermoregulation. Okay, we know that liver is the largest metabolite organ, all the metabolic activities occur in liver, right? So obviously heat will be produced, so this organ works as a thermoregulator. So thermoregulation also occurs in liver, right? Next we have that production of plasma protein. some clotting factors. For example, albumin, prothrombin, albumin, and fibrinogen. So basically these are some plasma proteins which are produced in liver. Next we are having bile. Now we have its secretion which is known as bile. Basically this bile contains some bile acids. For example, cholic acid, D-cholic acid, D-cholic acid. Quino-D-cholic acid. Quino-D-cholic acid. So basically these are some acids which are present in bile. Next we are having some bile salts. Bile salts which are also present in liver. For example, sodium hydrogen carbonate, like sodium bicarbonate, NaHCO3, right? This is a salt which is present. Next we are having sodium taurocolate. We are having sodium taurocolate, sodium glycolate. Right, these are basically some salts which are present in bile. Basically these both salts help in emulsification of fats. Sodium taurocolate and sodium glycocolate, like they have the function in emulsification of fats, like this sodium taurocolate, and like these bile salts attach with fats in order to get dissolved in cell membrane. We will discuss this thing later on in my videos, right? Don't worry about this. Next we are having, like, the pH of bile is 8, right? Next it also have water, and it has some pigments. Bile pigments that are bilirubin and biliverdin. Basically these both are the pigments which are present in bile, right? Next we have, okay, basically we know that in spleen is the, like we can say, is a graveyard of RBCs. It is graveyard RBC, right? So here what happens, the RBC death occur over here. So in the result of death of RBC, actually they contain, like, some pigments are found which are known as bilirubin is formed by the death of RBC. Then this bilirubin is transported into liver, and that's why liver contains this bile pigment. And because of this bilirubin, like the presence of bilirubin, our feces color is brown, because of just this presence of this bilirubin pigment, right? So I hope this is clear to you. If you have any kind of questions, you can ask me in the comment section. I'll be glad to reply you guys, right?